Now, I hate to say I told you so, but I've just said it. And while I always knew there was much potential for using CRISPR activation for cellular reprogramming, and now we have a paper that suggests in more detail how and that it's very effective. So hello and welcome to the Sheekey Science Show, where in this video we'll take a look at this paper. CRISPR activation enables high fidelity reprogramming into human pluripotent stem cells, and a deep dive into CRISPR, CRISPR activation, and also cellular reprogramming. So firstly, before we go any further, we just need me to explain what cellular reprogramming actually is. And well, let's go back to 2006, when Shinya Yamanaka published the now landmark study whereby the introduction of 4 factors, the Yamanaka factors, OSKM, which when expressed in differentiated mouse cells, could reverse them to pluripotent stem cells. These are cells that have the potential to divide continuously, but also to differentiate into different cell types. So they were termed induced pluripotent stem cells. And so this is where the reprogramming comes in, because you reprogram a cell's identity. Now, I've made a few recent videos on cellular reprogramming, but I think it's important to reiterate why there is much interest around it. And there are 3 main reasons. Firstly, for disease modelling. For example, if you wanted to study neurons of an individual, instead of taking their actual neurons, which would be quite challenging to do, you could instead derive them from a stem cell and get that stem cell from one of their skin cells using this reprogramming technology. Secondly, you can use it to understand the biology of cell states and also cellular plasticity, how easy and difficult it is for cells to be reprogrammed. #biochemistry nerdiness. And then lastly, for regenerative medicine approaches. So the common method of reprogramming is giving cells the genes encoding the reprogramming factors such that they are overexpressed. However, having this forced expression of ectopic genes can cause the following problems: off-target gene activation, so expressing genes that you don't really want to be expressed, which potentially could be due to having a high concentration of these reprogramming factors that may bind to DNA where they wouldn't otherwise. Secondly, it can result in heterogeneous reprogramming, and that can result into different outcomes besides having a stem cell. As the best case scenario when you're doing reprogramming is to, achieve reprogramming, but you may also cause a cell to die. You may cause the cell to become senescent, or it may become transformed and cancerous. Not so good. But why does this happen, and is there any way to make this more controlled and safe? And so on from this, the cells that do become stem cells may be aberrant. So what is normal cellular reprogramming, and how does CRISPR help? Well, once these Yamanaka factors are expressed, the assumption is that they will then activate other genes, and the downstream consequence of these events results in a cell changing its epigenetic landscape and stabilising into the stem cell state. So what would be desirable features for reprogramming technology? Well, I would like a reprogramming technology that matches my personality: reliable, efficient, safe, with a good sense of humour. Okay, we can scratch the good sense of humour. But anyway, reliable. We want to know it's going to reprogram and not do something weird, e.g. not die or become senescent. And also, we don't want the cell to try, get halfway and go,
"That's it, I'm not going."
You want it to fully commit. And I suppose tied into this is you want it to be reproducible. You want it to do the same thing every time. And then efficiency. Reprogramming can take time. The cells need to make some serious changes. You might think slow and steady is potentially the best approach. You can't rush great art. But maybe a longer duration also means the cell is more likely to go off track. So maybe shorter is better. And then safe, which given my recent bike accident, this probably isn't a personality trait of mine, though I do sometimes wear my lab coat. Anyway, safety goes back to what I was saying with reliability. We don't want the cell to become uncontrolled and cancerous. We want to know that it's trustworthy. So CRISPR activation. Now, almost a year ago, I told you about CRISPR activation, even though it's been around for much longer than that. But essentially, it's a modified version of CRISPR. So conventional CRISPR, you have a guide RNA that recognises a specific site on DNA, and then you have a protein that binds that guide, Cas9, Cas9, this protein, has the activity to cut DNA and make a double-stranded break. You can then change the sequence of the guide RNA and target the CRISPR complex elsewhere. In CRISPR activation, we mutate Cas9 so that it doesn't cut DNA, but it retains that site-specific binding activity. And also you can tag Cas9 with other small proteins that promote transcription, so expression of genes. And so what would happen is you target Cas9 upstream of a gene you want to express, so let's say the Yamanaka factors, and it can activate gene expression. So that's why it's called CRISPR activation. So the key difference is that this is so-called endogenous expression, not ectopic. And it's actually been done already before using the 4 Yamanaka factors, so OCT4, SOX2, KLF4 and MYC, plus Lin28A, and a motif expressed near genes expressed in early development that they call EEA. But the thing was, this CRISPR activation approach had pretty low reprogramming efficiency, so not really fulfilling my personality checklist. So this brings me on to the new publication, where they conclude that these findings support the use of CRISPR-A for high-quality, pluripotent reprogramming of human cells. So how did they do it, and are there advantages or disadvantages to the CRISPR approach over these more conventional ectopic gene expression approaches? So to speed up their discovery process of other potential target sites, they used a system that enables reprogrammed cells to be easily detected and analysed. So their approach involved using cells that grow in suspension but then attached to the surface of a plate when reprogrammed. By doing that, they identified the most efficient reprogramming when they had extra EEA targeting and when they targeted the promoter of MIR32367. This latter thing is still a gene, but instead of coding for a protein, it codes for a microRNA. I don't have the time to go into all the details as to how these are expressed or why they work, but I did make a video a few years back with more details if you want to learn more. But like with the activation of the Yamanaka factors, which are transcription factors that can regulate the expression of multiple other genes, microRNAs can also influence many different genes, and so you get much bang for your buck. But one of the rationales for these specific microRNAs is that they reduce the expression of some factors that repress reprogramming, so you're repressing the repressors. Anyway, with this extra EEA targeting and a microRNA targeting, they called their technique CRISPR-A plus me. It's getting very personal today, but me here stands for the microRNA sites they're targeting and E for the EEA motif. Anyway, that was a cancer cell line, but they also showed that it worked in human fibroblasts, and CRISPR plus me was the only reprogramming condition that properly induced pluripotent stem cell colonies from fibroblasts derived from an 83-year-old man. And this is a cell line that's known to be difficult to reprogram. And so the cool thing that they also did in this paper was that they did single-cell RNA sequencing to see the gene expression profiles of the cells using this approach compared to a transgenic approach. What they observed based on the gene expression was that mid-reprogramming, so day 15, the CRISPR-A plus me cells looked more like the reprogrammed cells than the other transgenic approaches, suggesting that the kinetics of reprogramming were also being enhanced. So that's how, but what are the advantages of using this CRISPR-A approach? Well, as I mentioned earlier, you're expressing the endogenous genes, and you're modifying the epigenetic landscape in situ. And by having this endogenous instead of exogenous expression, you also retain extra steps of gene expression such as splicing and regulatory elements, which may also influence the control over the expression of these factors, which might be useful from a safety perspective. And then the second thing with using CRISPR in general is that it enables high multiplexing. Once you've got Cas9, you can just add multiple guide RNAs for different sites that you want to target. So this is the magical summary figure. You start with some differentiated cells, add the CRISPR machinery targeting Yamanaka plus more EEA and microRNA 302, and you get induced pluripotent stem cells. I think there's many studies that could be done to further explore this technology. I think it'd be kind of cool to see where, if they could better map out the chromatin landscape of these cells and also see potentially why the transcription factors, the Yamanaka factors, are binding in these cells compared to the other approaches, to further understand, what is it about this approach that seems to be more effective. And then they can link it back to their transcriptomic changes. But anyway, I'm digressing. So, can CRISPR solve aging? Well, I guess we'll have to wait and see. But whether it's through CRISPR activation or other CRISPR techniques I've not mentioned in this video, such as gene editing, knockout screens, or using alternative Cas proteins for diagnostics, it will certainly aid in our future understanding of the aging process and how best to treat and diagnose for it. So with that, I'd like to thank myself for making this video and all the cells involved. Thank you to my Patreon supporters, and thanks for listening.